Hello, my name is Chelsea Titus, and I am a master's student at the Savannah River Ecology Lab. Before we begin, I'd like to give a big thank you to my co-authors, Courtney Pierce, Dr. Timothy Smyser, both at the Wildlife Services at USDA, Dr. Stephen Webb at the Noble Research Institute, and my advisor, Dr. James Beasley at the Savannah River Ecology Lab. Today, I'll be talking to you about the age-old question, what is a sounder? Specifically, the genomic relatedness of wild pig social groups. Wild pig social groups are typically thought to fall into two categories. Female sounders, pictured in the top right, which are observed as a group of breeding age females of variable size with their offspring. These females are assumed to be closely related family members. The second category is solitary breeding age males, which is represented in the picture on the bottom right. Reports of size and composition vary greatly across the U.S. Sounder observations have ranged from a single female with piglets to as many as 28 females with piglets coordinating together. Males are also sometimes reported in groups, but this is thought to be rare. Territoriality and avoidance varies greatly across studies, and there have been some observations of female dispersal also thought to be rare. Genetics has long since been used to investigate mammalian social systems. Such studies have recently shifted towards genomics for better resolution of complex and variable systems. There has been little genetic investigation to confirm social group composition across the invasive range. And among these studies was low sampling and little consensus, and genomics is underrepresented in the literature. Genomics has recently been used to elucidate the complex history of wild pigs across the US, which we can see in the figure to the right from Smyser et al. 2018. 3 genetic lineages were found to contribute to the rise of modern wild pigs. The Western heritage breeds at the top contribute in higher proportions throughout the range than commercial breeds at the bottom. However, European wild boar can be found to contribute high proportions in isolated populations across the US. There continues to be a critical need to incorporate modern genomic techniques in studies of wild pigs, especially their social structure. I hope to contribute to this with 2 objectives. Describe the genetic relatedness and demographies displayed within wild pigs social groups using large data set and collected across nearly a decade, and then compare trends in social group compositions between 2 study areas that are dissimilar and isolated from each other, which are represented in the figure below. My first study area is Savannah River Ecology Lab, or Savannah River Site, at Aiken, South Carolina and Georgia, represented in the shaded area of the map on the top right. Pigs here were captured from 2012 to 2019 on the Savannah River Site. The presence of the pigs have predominated DOE construction, and the area is surrounded by a permeable border. Habitats include hardwood savanna forest, pine-dominated forest, and swamplands. Recently, it's been estimated to have as many as 5,000 wild pigs. Oklahoma is my second study area, and pigs captured here were from 2010 through 2017 as part of NRI's research ranches. Presence of wild pigs predominates NRI's management, and habitats include heterogeneous wooded and prairie areas, where wooded areas are predominantly oak, elm, and hickory, and prairie areas are predominantly blue stem, switchgrass, and Indian grass. And I've represented this area roughly with this yellow circle on the bottom left map. Genetic samples were collected as part of an ongoing research and control collaborations. Whole groups were caught with corral or drop nets, and samples were taken from necropsies, hair, and ear punches. These samples were then sent to the National Wildlife Research Center at the USDA APHIS. There, individuals were genotyped for 62,331 single nucleotide polymorphic loci, also known as SNPs. Genotypes were then filtered for quality control using the SNP relate package in R, and this was to ensure the most informative SNPs for the study. We were then left with 9,306 SNPs for analysis using the R packages SNP relate and Genesis. SNP relate, we calculated pairwise measures of King robust kinship coefficient estimator, and with Genesis, we adjusted genetic relatedness, or R, in the presence of potential family or population structure. Individuals were then assigned to social groups based on having the same capture location, same capture date, and being captured as part of a group with one or more breeding age individuals. We then removed piglets and small juveniles, that being females less than 70 pounds and males less than 100 pounds, and we also removed large breeding age males that were found to be unrelated and calculated as a separate group R. These groups were designated with the same female group number followed by a -M. Furthermore, I assigned general degrees of familial relationships to the pairwise comparison using Manichai Cole et al. 2010's R ranges associated with the familial degrees from their 5,000 SNP analysis. For results, I'll start with SRS and then move on to Oklahoma. SRS, we had 292 individuals assigned to 116 social groups. To avoid flooding or diluting relatedness, we excluded those non-breeding age individuals and unrelated breeding age males, which we can see in the graph. On the left, from left to right, we have the smallest group size to the largest, represented by this single peg here for one individual, and then this group of pegs here on the right for the large 14 group here. On the left axis, we have the group size, which is for the bars. Blue is the original group size, the original number of groups for that group size, and orange being the new number of groups for that group size after the males and unrelated breeding age males and non-breeding age individuals. On the right axis, we have the average relatedness, or group R, that follows the line graphs here, the blue diamond and the orange square. As you can see, before and after the exclusion, there have been changes in the relatedness and number of group sizes. Moving on to my favorite graph, we have the compositions of each group represented. The top figure is the sex composition, with females being red and males being blue, and the numbers being the count of individuals. On the y-axis is the percent composition, and the x-axis is the group IDs. The next figure is the percent composition of age classes, and below that is the percent composition of relatedness. After filtering out suitor males and non-breeding age individuals, the most common group size was 2, and as you can see from the top figure, it was typically females, and these females, according to the bottom figure, were typically first-degree relatives. However, some groups displayed unrelated interactions, which is represented by these gray shaded areas. The two graphs are gradient colored, so the middle graph, the darker the color, the older the representation, and in the bottom graph, the lighter the color, the more closely related the representation. Groups were most likely to be composed of just adults, which we can see in the middle graph here, but many also included other age classes such as subadult, yearling, and juvenile. Male-only groups on the right side were more likely to be composed of a single age class, typically yearlings of first or second degree relatedness. In general, groups display just first degree relationships, which we see in the bottom graph here, but second degree only and in combination with other degrees of relatedness were also common. It was not uncommon to have a group that included unrelated interactions, which we once again see in these gray bars. 4 groups were composed of only unrelated, typically older individuals. As for the Oklahoma results, we had 96 individuals that were assigned to 43 social groups. And again, we excluded non-breeding age individuals and unrelated breeding age males. Unlike SRS, there were large changes in group size and group R, which we see in this graph here. This is the same graph as the SRS graph, but for Oklahoma. You can really see the changes. The largest group size here was 42. However, that was removed when we excluded the non-breeding age individuals, and the number of solo individuals greatly increased. Once again, we have the percent composition graphs with the sex composition on top, age composition in the middle, and degrees relatedness on the bottom. The top graph has the proportion of females in red, proportion of males in blue. The middle graph has a proportion of adults in dark green and getting lighter as they get younger. Bottom is the proportion of degrees relatedness with the proportion of first degree relatedness in light blue and getting darker as they become more unrelated, more distant related. And for all of the figures, group ID is on the x-axis. After filtering out suitor males and non-breeding age individuals for this set, the most common group size was also 2. However, one group was caught with as many as 42 pigs before filtering out those unrelated individuals, or unrelated males and non-breeding age individuals. Our female-only group on the left half of the graphs were typically the same size as the male-only groups on the right side of the graph. However, there were a couple that were larger, which we see 6 individuals here and 4 individuals here. Groups were typically composed of just subadults or in combination with juveniles, which we see in the middle figure here. Subadults being this moderately dark green and yearlings being this moderately light green. Groups were typically composed of multiple degrees of relatedness, and this is usually included a majority of first degree relationships in the light blue here. Groups that included at least one male, which we see towards the right half of these figures, were more likely to display some unrelated interactions. And keep in mind, this is after we excluded unrelated males. This unrelated proportion was typically 10 to 40%. Groups were typically related and breeding age females, like we previously believed, with their offspring. However, the extent varied between study areas. Prior to excluding non-breeding age pigs and unrelated breeding age males, SRS's largest group was 14 individuals, whereas Oklahoma's was 42. After exclusion, these group sizes changed for many of the Oklahoma groups, but not many groups changed for SRS. Group relatedness SRS range did not change much across groups, but almost all of the group's relatedness changed for Oklahoma. Oklahoma females had more variation in the proportion of each degree of relatedness, and more groups included third-degree relationships than SRS. This indicates distant relatives associating within the group. Relationships displayed in groups became more complex when males were present for both SRS and Oklahoma, even though unrelated breeding age males were excluded from the analysis. Here is an example of some of the complex relationships, going from moderately complex to more complex, and keep in mind that the pictures are not of the actual sounders. First up is group 32 from SRS, where we have a subadult female represented by this circle, and an adult female represented by this circle, and the male represented by this square. And he is an adult. So females are circles, males are squares. The thicker the line connecting the two, the more related they are. If there's no line connected, then they are unrelated. In this first group, 32, we have the subadult, the 2 females that are second degree relatives, and the adult female is a third degree or distantly relative to the adult male, but these two are not related. Moving on to group 24 in the top right, we have an adult female who has a first degree relationship with a juvenile female. And on the other side, we have a juvenile female first degree to this juvenile female. However, these two groups, even though they were caught together, are unrelated. It is likely that the 2 juveniles related to each other had their mother outside of the trap or off foraging or something. The bottom group from Oklahoma, 53, is a much more complex situation going on here. So we have an array of first-degree relationships here, where this yearling, 132, is related to these three, but not related to anyone else. We have 128, this juvenile male, related to 126 juvenile male, but not related to anyone else. And then we have this juvenile male 129 who's related to 126 and 131 distantly. And he is the missing link between this group of first-degree relationships. These three juvenile males are all first-degree relatives to each other, but not related to anyone else but 129. It gets even more complex when you start adding more individuals. And so keep in mind that circles are females, squares are males. The thicker the line connecting them, the more related they are. And if there's no line connecting them, they're unrelated. And this just becomes a huge hodgepodge of relationships in this group. Very complex and very dynamic associations here. Oklahoma groups were generally more likely to display complex array of degrees of relatedness than SRS. However, SRS groups were composed of high proportion of first and second degree relationships. Oklahoma groups had smaller proportions of these degrees of relatedness and more often displayed third degree relationships. This may be due to differences in size. However, group size may also be influenced by other factors. Despite excluding unrelated breeding age males, both study areas had some groups with a proportion of associations classified as unrelated. SRS had more groups with higher proportion of unrelated associations in Oklahoma, including 2 female-only groups and 2 male-only groups composed of completely unrelated individuals. Oklahoma only had 1 completely unrelated group, which is 2 unrelated breeding age males that were caught with an unrelated breeding age female, group 75 and 75M respectively. Differences in relatedness between SRS and Oklahoma may be due to the differences in group size, habitats in the areas, climate of the area, resource availability, or even the historic genetic lineage. In conclusion, wild pig social groups are more complex than previously believed, and the level of complexity differs between the 2 study areas. While SRS social groups are likely to be 2 to 3 closely related older females and their dependent offspring, wild pigs in Oklahoma were likely to include 3 to 4 older females of dynamic relatedness and their dependent offspring, with the potential for Oklahoma groups to be much larger. Both areas included unrelated pigs associating with each other, but SRS is more likely to have a group of only unrelated individuals. This may suggest a greater willingness to form new groups and associations and may be influenced by the differences in resources or attributes across the different landscapes, as well as the historic lineages that contribute to the different populations that I mentioned in my introduction. Moving forward, I hope to further resolve these associations within groups in the 2 areas, as well as explore relatedness between groups in each area. And with that, I would like to thank the many people that's helped me on this project, as well as my funding sources, UGA, DOE, USDA, ACUS. Thank you.